Hi, I'm Mimi Gerner, and here we are in the GAM Labs Tissue Culture Room at the University of Wisconsin-Madison. I'm going to show you around the room so you can see some of the things that we use every day. Over here we have our water bath, where we warm up all of our media for the cells. We have our microscope attached to our computer, where we can look at our cells but also have the ability to take pictures of our cells. Over here we have the incubator, where we house all of our cells. This environment allows the cells to stay warm in a controlled gas environment and also in a very humid environment, which they thrive in. And here we have our refrigerator. This is where we keep all of our media. It stays nice and cold in here so that it stays fresh for the cells. These are our Biosafety Cabinets. They allow us to work in a sterile environment with our cells. Right now the UV light is on, which is killing any bacteria on the surface of the cabinets. When we open them up, you might hear a blower turn on, which keeps the airflow moving and keeps the outside air from entering into the sterile hood. You might even hear the air ventilation flowing through the room, which is important to keep the air in here as clean as possible. So I'm going to get out some cells from our incubator for us to look at. This is something we would do every day. We take cells out of here, check for differentiation, or to just check on their overall growth. So I'm going to take out some human embryonic stem cells and some other cells that have already started to differentiate. Those are the ones in the flask. So I'm going to place these cells under the microscope. I'm going to look at the plates first to check for differentiation, and then after that I'm going to look at the flask to see how the differentiated cells are coming along. Right now I'm focusing the cells, looking them over, checking to see if there's any differentiation on the cells, which we want to remove. We want the cells to stay in an undifferentiated state so we can continue to grow them and perpetuate them. So if we do find differentiation, we have an objective marker that just works like a little stamp. So we would find the differentiation in the well, switch the objective to the stamp, and then by lifting up on it, we mark where the differentiation is. Then we can take this plate into the hood and scrape off the differentiation with a tool. So by lifting up on the marker, we make a stamp, and that leaves a spot that marks where our differentiation is. So here's another look at what I'm visualizing on the microscope. Here we have a small stem cell colony that's in an undifferentiated state. You can tell it's undifferentiated because it has a nice clean border, a nice round shape, and the cells are still pretty small and tightly packed together. Around it you can see mouse embryonic fibroblasts, which are on the plate in order to support the stem cells. Here's an area of differentiation that I'm going to mark and remove. You can tell there's differentiation here because there's not that nice clean round border. The cells are starting to spread out, get more far apart. There's also this area of a large cluster of cells that are actually growing off of the plate instead of just being one flat layer. You can still see the mouse embryonic fibroblasts behind it, but here we can see all the differentiation happening. I'm going to go ahead and mark this with my marker. We're going to lose the image for just a second while I switch the marker into place and make my stamp. And now you can see the blue border that marks all around the area that I'm going to remove once I get the plate inside of the sterile hood. So now we're looking at something called embryo bodies. These are cells that have started on the pathway to differentiation. These were cells that were lifted off of the flat surface of a plate, and because they were lifted off, they balled up into these spherical shapes that we call embryo bodies. Now, these aren't embryos, but they do have some similar characteristics, such as they are starting to differentiate and they're a ball of cells. But that's about where the similarities end. So these balls of cells aren't pigmented, they're not really brown. If you were to look at them with your naked eye, they would look a whitish creamy color. Here they look so dark because the light just can't get through them because there's so many cells in each mass. So I'm going to get my media out of the water bath. It's been warming in here for about 15 minutes, up to a temperature close to 37 degrees Celsius. 37 degrees Celsius is the human's body temperature. So we want it to be as close to this temperature as possible when we feed it to the cells. because they're human cells. They want to be at the normal human temperature at all times. So now I'm at my hood. The UV light's been on for about 15 minutes while the media's been warming in the water bath. The UV light, like I said before, is killing any bacteria that's on the surface. We're going to spray it down with a 70% ethanol solution once I lift the sash up. This ethanol solution is going to further kill any bacteria that's inside of the hood. It's really important that we keep this area sterile because the cells inside of the dish don't have an immune system to protect them like they would if they were a growing embryo inside of a uterus. So I just got my cells out of the incubator with the spot that I marked. First thing I'm going to do is spray these down because anything that I bring into the hood has to be sprayed down and be as sterile as possible. I'm also going to spray down my media bottle and put that in the hood. As well as my hands, because they're going in too. So I'm going to remove the cells that I marked just by scraping them with the pipette tip. And the cells are so small you can't see them, but the ones that I scraped are now floating in the media. The ones that are undifferentiated are going to stay stuck down to the plate. I'm now going to aspirate off the media. This is going to remove any dead cells that are floating in the media, the spent media that the cells have used all the nutrients out of, as well as the cells that I just scraped off of the mark area. Those will be removed in this step also. So I'm going to remove the lid, angle the plate up toward me so that the media goes down to the bottom of the well, and this little vacuum apparatus takes the media right up. I'm going to remove it from both plates and then use a different pipette to replenish the media back into each well. So now I'm going to get my sterile pipette in its sterile wrapper and using this pipette aid, attach it to here, being careful not to touch any of the sterile parts on the pipette. I'm going to go into my media bottle, take up only the media that I need, which is 2.5 milliliters per well, and deliver 2.5 milliliters of media to each well. Now that the cells are fed, they're ready to go back into the incubator. And here they will sit until tomorrow.